Tonight's speaker again is Rob Martienssen, professor at Cold Spring Harbor Laboratory in plant biology. For those of you who may not know, Cold Spring Harbor Lab is based on Long Island, and the scientists there conduct cutting-edge research on cancer, the brain, plant biology, and historically much of their research has centered on genomics, DNA. DNA, as you all know, is the blueprint of life, of every form of life. In every cell, there's a map of your body plan, your growth and development, your inherent strengths and weaknesses, perhaps, and maybe your disposition. DNA is a complex code that we're slowly cracking, but it turns out it's even more complex than we had initially thought. Genes don't always just stay in one place. They get turned on and off, and sometimes they even jump around. Enter Dr. Rob Martienssen. Dr. Martienssen is at the forefront of research in the field of plant genetics and epigenetics. In addition to directing the plant biology lab at Cold Spring, he's a fellow of the Royal Society, and recently he was named by the Howard Hughes Medical Institute and the Gordon and Betty Moore Foundation as one of the nation's 15 most innovative plant scientists, and he was chosen to join a new initiative that will intensely focus on fundamental plant science research. The potential applications of research in plant genetics and epigenetics are very stunning, actually. Better biofuels, higher yielding crops, perhaps even the long sought after black tulip, something that I've been searching for my entire life. Oddly, yeah. Please welcome Dr. Rob Martienssen.
That was quite an introduction. It's just such a pleasure to be here. It's a magnificent place. I'm expecting to learn a lot. The first thing I've learned is the next time I give a seminar at a symposium, I want an open bar. Spectacular idea. All right, so I'm going to tell you about plant biology, but my theme is going to be something that's very familiar in plants and is becoming more and more familiar in biomedical research, namely clones. Clones, of course, inherit the complete genetic complement of the parent organism and are therefore genetically identical, and they've caught the imagination of the media over the last few years, including the movie industry, of course, from everything from the boys of Brazil to this guy. And of course, cloning, reproductive cloning, as this is imitating, has become very controversial for all sorts of reasons in biomedical research. But I want to persuade you that clones are actually a very natural part of our planet, especially in plant life. And in fact, the word clone is derived from the Greek word for a trunk or a branch, klonos. And it refers, of course, to something you've probably, well, maybe not in Brooklyn, but you've probably all done occasionally, which is to take a cutting from a plant and grow an entire plant from it. That is a clone. And this was recognized, of course, many thousands of years ago. And in fact, clones are perfectly natural. It's not just gardeners who make these things. And in fact, the very largest known organism on the planet is in fact a clone. It's a stand of quaking aspens in where is it again? In Utah, in Fish Lake, Utah. And this stand is thought to be, the roots of this stand are thought to be something like 80,000 years old. The roots are all connected, and there are something like 50,000 stems that have clonally grown from this theoretically, genetically identical organism. This organism weighs in at around 6,000 tons, metric tons, and covers many hundreds of hectares. So this is just a single organism that's been around for a very long time. Interestingly, of course, because it's a clone, it's all the same sex. It's actually a male clone, and has been around without the benefit of female clones for a rather long time. It was given a name by the botanists who discovered it genetically. They call it pando, meaning I spread in Latin. And as I said, it grows from its roots in this clonal fashion. Other clones are extremely important in agriculture and horticulture, as I'm sure you're all aware. One that might be of more interest to this audience are, of course, grapes. Grapes are clones. They're highly prized clones, of course. And they're grafted onto rootstocks in order to maintain them and actually maintain them free of various pests. what people may not realize is these clones are literally hundreds of years old. The most important Pinot Noir clones, for example, were actually derived in the Middle Ages, and they've been propagated in this way ever since. Now, of course, one reason to use a clone is because it's genetically identical not only to all the other clones, but to the previous clones as well. At least that's what you would expect. In fact, that's not always the case, and some of our favorite grape varieties, although they are prized for their uniformity, this is actually Pinot Noir, I think growing in Chile, they also throw off sports, and these sports can be extremely important. So, for example, Pinot Blanc is actually a derivative of Pinot Noir. It came from a clone. The clone was supposed to be genetically identical, but in fact it wasn't. And the reason it wasn't is because there are, if you like, epigenetic mechanisms above and beyond genetics, which can change plants, and for that matter animals, in a way that doesn't depend on sexual reproduction. It doesn't depend on meiosis or the mixing of genes through sex. And in fact, the reason for these particular varieties was a phenomenon that was originally discovered, I'm pleased to say, at Cold Spring Harbor about 50, 60 years ago now, which is namely transposable elements, sometimes called transposons. And these are really one of the prime causes of these epigenetic changes that we see in nature. This is actually a picture taken from an ear that, we call them ears, not cobs, by the way, these are maize ears, that was actually saved by Barbara McClintock, who discovered these transposable elements about 60 years ago, and we took a photograph of it recently. It's in perfect condition. And what it shows is that color genes, which give you that purple color, rather like Indian corn, are changing within the kernel. So the gene is turning on and off in these patterns of speckles, which are very striking, and which actually follow certain genetic rules if you do manage to cross them and so on in a genetic study. So Barbara was able to recognize that transposons were jumping genes that could move around the genome, even in the absence of recombination and meiosis. They could actually move themselves around the genome. And when they land within a gene, they will change the nature of that gene and, for example, change the color of corn. And in fact, it was one of these transposons that changed the color of wine from Pinot Noir to Pinot Blanc. And in fact, this transposon, which I've illustrated, I should have a pointer here, maybe I can try this, maybe it won't work. The normal wild type Pinot Noir at the top there has this gene, it's called VV-MIB1, doesn't matter what it is, that causes the red color. When a transposon, a full-length transposon, inserts near that gene, it abolishes that color, and now you get Pinot Blanc. But then if the transposon moves again, leaving just a bit of it behind, now you get a sort of gray color, and this is Pinot Gris. So each of those clones was actually derived from a parent clone in a way that didn't depend on sexual reproduction. It happened because of the action of these transposable elements. It happened for an epigenetic reason, if you like. Well, why are clones so successful? We always think of clones perhaps in a rather derogatory way, but in fact clones are successful in nature, the oldest living organism, and so on. It should be said that they're not successful in all respects because they don't need sexual reproduction. They often sacrifice fertility. As I mentioned, Pando probably hasn't had sex in about 10,000 years. And in fact, the pollen count is very, very low now in that stand, and it probably has decreased over all of that time. But of course that doesn't necessarily matter because it's a very successful growing organism. And part of the reason that it's successful is that it has maintained something called heterozygosity. Now that's a long word, but it's a very simple concept. And that is that it's that in the original sexual cross that produced the very first root and the very first plant, it received genome complements from both its maternal and its paternal parent. In other words, mother and father. And that combination is a hybrid combination. And hybrid combinations are usually better than inbred combinations. And what clones can do is retain that hybrid combination throughout their generation of new clones. The clones are genetically identical, so they retain that hybrid vigor. Now, if they went through sexual reproduction, the mother and the father's genes would be randomly selected in each generation of sexual reproduction, and you very quickly, in fact, you'd immediately lose that hybrid vigor in the next generation. So this is why hybrid vigor is so important, and this is why clones are successful. Sexual inbreeding, on the other hand, if you were trying to maintain the genetic constitution of something by simply crossing it to its siblings all the time, then you would end up with something called inbreeding depression. And in fact, that's known in humans. Just think of any of the British royal family, for example. It was, in fact, in corn that George Schull, working again at Cold Spring Harbor, figured all this out, and he did so in a really remarkably intuitive way. And I recommend, I'm not going to tell you exactly how he did it, but I recommend if you can see that website there, there's a Cold Spring Harbor website called Weeds to Wonder, Weed to Wonder, I'm sorry, which actually has a very nice video of George Schull explaining hybrid corn. But basically what he did was he inbred corn so that they were completely homozygous. And of course they were pretty sick, or at least had relatively small yields. But when he crossed two inbreds together to make them heterozygous, now he got hybrid corn, which was extremely vigorous, and has actually been the basis for the corn seed industry ever since. This was done about 100 years ago, and corn yields have gone up, and I always forget the number, but it's something like tenfold since then, largely because of this insight. I'm pleased to say that it's not just corn that benefits from this, and this is a picture of a colleague of mine at Cold Spring Harbor, just a good picture really, but Zach Lippman, who has been working on high-yielding tomato varieties and has discovered that hybrid vigor can be reduced to just a single gene. This is a gene that actually came from a wild tomato species, and if you make a heterozygote that has one functional copy of the gene and one non-functional copy of the gene, that plant will actually yield a lot more tomatoes than either of the homozygous parents, which only had one type of gene. So this is hybrid vigor. This is why clones are successful. But of course, not all plants are clones. And of course, we want to start using plants in many different ways. We just heard in the introduction. Of course, for food, and corn yields over the last 100 years have made a really big impact on that. I should say that hybrid rice and hybrid wheat are much more difficult to make, but they're also beginning to have a major impact as well. But in the future, plants are going to be asked to do an awful lot more than just feed the world. And I probably don't need to lecture you on climate change and biofuels and all of that, but I'll just give you a quick summary of where we stand. Basically, fossil fuels represent millions of years of the accumulation of biomass from plants, and if you talk about coal, actually mostly from animals in the case of oil, but nonetheless millions of years of accumulated biomass. This, of course, is being rapidly burnt up in various ways to generate energy. But of course, a byproduct of that energy almost inevitably, is atmospheric carbon dioxide and other greenhouse gases as well. And these, of course, are having a dramatic and rather drastic effect on climate and have done since the beginning of the Industrial Revolution. So the idea of biofuels, of course, is that atmospheric carbon is fixed by plants. That's why we're able to use fossil fuels in the first place, is because millions of years of plants and animals in the past have accumulated carbon in this way. If we can then use existing plants rather than fossil plants as biofuels, then we get energy without a net carbon increase. And this is the whole idea of biofuels. So over the last few decades now, potential plant sources of biofuels have been investigated a great deal, and some of them have actually been used to some success. And the way we measure that success is as something called energy balance. So what this means is that even if you have a highly productive biofuel plant, of course you have to grow it, harvest it, fertilize it, transport it, usually in a truck, to a plant somewhere where it'll be turned into a biofuel, either ethanol or biodiesel. And of course all of that takes energy. And so the energy in and the energy out is the energy balance. Now many of you will be familiar with corn ethanol. You probably don't have cars, do you, in Brooklyn? But anyway, if you had a car, then you would know about 10% ethanol or even 15% ethanol being a requirement these days. Most of that ethanol in the US comes from corn, from maize. Unfortunately, the energy balance of corn is very poor. This is actually an estimate, 1.5, which means you get 1.5 units of energy for every unit you put in. In fact, that's an optimistic estimate. Some people think it's more like 1.1. So you might be wondering why you do it at all. Well, in fact, liquid transportation fuels like ethanol have a high premium value to the economy. So even if you can make electricity some other way, you're still interested in having liquid transportation fuels. However, there are other plants that do better. For example, the three listed on the top, which I'll talk about later on. Interestingly, these are all propagated as clones. But before we get there, I'm going to explain the difficulties of trying to get corn to be a better biofuel.
Do you have the figures for hemp? How does hemp compare with those other?
I'm sorry, with what?
Hemp.
Hemp. Oh, right. So
Yeah.
let me. The nearest thing I've got there is cellulosic ethanol. So it's not hemp in this case; it's actually miscanthus, but it's a similar sort of idea.
You have figures for hemp?
I don't have them. But cellulosic ethanol, I'll get to that later on a little bit. It's an exciting prospect, and who knows, hemp's got to have some use, surely. A byproduct. So the reason that it's hard to deal with these highly bred crop plants like corn is because agriculture is old. Farmers have been breeding corn for at least 10,000 years. At least that's one estimate. And similarly for other staple foods. And as a result, they've narrowed down the sorts of genes that are in these plants to the ones that we want. And I just want to give you an example of that from our work. This is a mutant of corn. It doesn't look much like corn, does it? But it is. It actually looks a lot like one of its relatives, sorghum, which can have an inflorescence a bit like this. This is the result of mutating just one gene out of the 50,000 in maize. And that one gene, of course, is present in every single maize line that currently exists in farmers' fields. And that, of course, is because you want, actually mostly for storage reasons, you want these beautiful straight rows of corn, double rows of corn. Did you know that there's always an even number of rows of corn? There are. The mutation, basically, the loss of this gene, gives you this perfectly viable plant, has perfectly viable seed, but it would never be used as a crop. And so this gene is identical, almost identical, in every maize inbred that's currently used. And so there's not the variation that you need necessarily to be able to drive a completely new purpose for this crop plant. Another way of trying to get at the sort of genes, the sort of genetic variation that we need for these new uses of plants is to take a leaf from the book of evolution. In fact, I think it was Dobzhansky who said that nothing in biology makes any sense except in the light of evolution. And I'm just showing you a picture here which actually comes from a wonderful collaboration we've had for several years now with the Museum of Natural History in New York and the New York Botanical Garden, and as well NYU. What we've done is we've looked at something like 200 different plant species spread right across the evolutionary tree. And we've gathered as much DNA sequence information as we possibly can. For many of them, that's the complete genome. For some of them, it's part of the genome. But by lining all these genes up, we can now put all these species on a tree and say how they were evolved. And we were able to do this just in time for the 150th anniversary of The Origin of Species, written by, of course, Charles Darwin, who's rather unflattering portrait from a cartoon from the 1850s. Is shown on there. And when we do this, we can find the evolutionary relationship of species that might provide some of the genes that would be useful in manipulating, for example, trying to breed corn to do something more energetically favorable. So, for example, sugarcane and maize are actually reasonably closely related. And if we figure out why sugarcane has so much sugar, we might be able to get maize to make sugar in its stalk. Completely useless for a farmer, who's trying to make crop plants for food, but very useful for bioenergy. Of course, we can also use these species directly. They haven't been bred very much. In fact, many of them have only entered breeding programs in the last few decades. But nonetheless, plants like sugarcane and oil palm actually have a lot of potential. However, again, because they haven't been bred so much, and because we typically want to use a hybrid, those hybrids narrow the germplasm a lot. So for example, sugarcane is actually a hybrid between two wild, naturally occurring plants, one from, if I remember rightly, Indonesia and the other from Africa, which when hybridized give you this very high yielding sugar. However, that's it. That's what sugarcane is. It's basically a clone of that original series of crosses. So there's not that much germplasm to play with from a breeding standpoint. But nonetheless, these plants are very energetic, and because they're clones, they have retained much of their original hybrid vigor. So here are the best energy crops so far. Miscanthus is a grass, which hopefully with a little more work, and it is going to take more work, we'll be able to convert into ethanol using something called cellulose instead of starch. I'm not going to get into that, but there's a lot of investment right now surrounding Miscanthus. Miscanthus is in fact a clone. It's a huge grass from Japan. It grows to about 18 feet tall. It's quite amazing. But it can only be propagated as a clone, as a cutting. And so actually to propagate a large amount of the Midwest, it's currently estimated it would take several hundred years to get that number of cuttings. But nonetheless, it works. Similarly, sugarcane is grown predominantly as a clone. Yeah, you're far too young to remember that one. So, sugarcane is propagated by just taking a cutting and sticking the cutting back in the field. You can do that for something like 6 or 7 generations. Then you have to go back to a seedling because it does eventually deteriorate. But nonetheless, the clonal nature of these plants has really helped boost their yields. And again, I think that this is a more optimistic result of reproductive cloning than some others from Brazil. So, yes. So inspired by this success of sugarcane and other sorts of clonal plants, there have been several attempts to make plants that are not naturally clonal into clones. And one of the most high visibility efforts to do that is the humble oil palm tree. This is a palm tree that was originally grown in Africa and actually did undergo some breeding efforts by farmers in Africa a few hundred years ago. But breeding, nonetheless, is very difficult. Trees, take an awful long time to grow. You basically have to leave your genetic experiments in your will, to your children, because it takes decades to see the results. But nonetheless, palm oil has a lot of potential, recognized, in fact, by Virgin Atlantic, who managed to fly a jet plane using fuel that was derived from palm oil. Well, over the years, people have realized that oil palms, in fact, this may have been realized even in Africa a few hundred years ago, but there's some evidence for that, or even earlier, that indeed hybrid palms were the most productive. And once again, rather like that example from Zach Lippman, it turns out there's a single gene in palms that controls the thickness of the shell. And if you look on the left of this diagram, you can see those are little fruits from the oil palm tree. And when they're cut in half, you can see something that looks like a miniature coconut stuck in the middle of that fruit. And that's actually what it is. These are closely related to coconuts. And that coconut has a shell. And that shell gets in the way of making oil from the surrounding tissue. These ancient farmers actually managed to find a strain of this oil palm tree that didn't have the shell. It's called Passifera. So you can see it had the little coconut endosperm in there, but not the shell. And it was realized very quickly that that was no good because the fruits would easily get rotten and the shell was somehow protective. And it was the hybrid that really made the difference. So these hybrids are what are grown in plantations of oil palm, and these are the plants that produce all the oil. So a few decades ago, a number of companies in Indonesia and Malaysia mostly decided to try to clone these trees. because if they just took a hybrid and allowed it to normally pollinate, it would quickly lose its hybrid vigor, because sexual reproduction would randomly pick one gene or the other, and you wouldn't always have a hybrid. So they tried to clone them instead. And they did this by taking cuttings, actually putting them into tissue culture to really multiply the number of clones, and you can literally get thousands of palm trees from a single hybrid in this way. And everyone thought this was going to be fantastic and the answer to all their hybrid problems. But it turns out, as I mentioned, the case in grapes, that in fact not all clones are equal. This was, of course, famously discovered in animals from Dolly the sheep, whose clones actually had specific diseases that were not present in the original, in the mother animal. And similarly, I sort of picked this slide because it looks a little like a sheep, but the oil palms that were generated by cloning, although most of them were okay, some of them, and sometimes many of them, were not. And they had abnormalities, for example, different fruit types, that were not found in the original mother palm. And the reason for this is, again, this epigenetic effect, this effect that occurs outside genetics, and in many cases is due to issues surrounding transposable elements. So if we're going to make successful clones, we're going to have to do it in some other way. As I just mentioned, so clonal propagation has a lot of advantages. It maintains hybrid vigor, but it has this epigenetic consequence, which, depending on the plant and the way you make the clones, can be very severe. However, there are plants that go through a form of cloning, reproductive cloning, that involves cloning through the seeds. Now, this means that all the normal activities of sexual reproduction happen, so you make a seed in the normal way, but that seed only comes from the maternal parent. And it hasn't undergone sexual reproduction, it hasn't undergone meiosis, there's no mixing of genes, and these seeds are much more stable than the sorts of clones that you can make in other ways. And some well-known examples are dandelions, for example, the little seeds that fly away, and actually St. John's wort, which has a number of other applications too. And these plants have clonal seed. So we know it's possible. Perhaps we'll be able to make clonal seed from hybrids such as oil palm or even corn. So how useful would these be? Can we make them? Well, yes, we think we can. I'm going to tell you some progress on that front, some very recent progress on that front. And just to sort of get to the bottom line before doing that, this could be a really big deal also for farmers in poorer countries who have a really hard time buying hybrid seed. Because remember, corn seed has to be made every summer. All those high school kids ripping the tassels of plants in the Midwest. Again, you've probably never heard of that, but in the Midwest this does happen. There are literally thousands of high school kids every summer ripping the tassels off, because that way you can make a cross in a very high efficiency and get that hybrid seed. And that hybrid seed is then sold to farmers for the next generation. But if you could clone that hybrid seed without having to do those crosses, then farmers all over the world could simply save some of the seed that they harvested and planted again the next year. Otherwise, you can't do that because of inbreeding depression, what George Shaw discovered. So clonal seeds are actually have a lot of value, not just in increasing yield, but also in allowing people to save their seed without losing it. All right. So in order to manipulate or breed clonal seeds, we have to understand something about the sex life of plants. And I noticed on the website for this that I was supposed to talk about the secret sex life of plants. Well, anyone who's ever left their car outside in the spring will know that it's not so secret. Right? I mean, there's an awful lot of pollen around. And in fact, there's even some pollen art I was able to find on the web, which I thought was quite spectacular. So pollen is obviously a part of the equation. It's the male germline of plants, and eggs coming from ovules and fruits are the female side. So in order to start looking at this question, we've been using a plant that's easy to use in the laboratory. It's small, which means you can grow it indoors. You don't have to have fields and fields of this stuff. It also produces a huge amount of seeds and a reasonable amount of pollen. and it's been used worldwide now by tens of thousands of labs. It's a plant called Arabidopsis, which is sometimes called Arabidabidopsis, actually, by people who don't like it. Its common name in England, for example, is called wallcress. You'll find it growing on patios and cracks between bricks and things like that. It's a very useful plant. It's been completely sequenced. We know every gene in the genome, and we can really try to figure out what's going on. So before I tell you about the mechanism that we think can help generate clones, I have to explain a little bit about genetics. It's going to be fairly short. You all know about DNA. What you may not know is that DNA, the maize genome, for example, the DNA found in every single cell in the maize plant, when measured end to end, is about a meter long. Okay, it's really long, and that whole thing has to be wrapped up in various proteins, in things called chromosomes, which end up in a 10 micron nucleus. So it's a 10 to the 5th compaction. So these DNA molecules are long, complicated, and how that packing happens is very, very important because it explains how the DNA is read during development. So when a corn plant decides to make a leaf or a root, it has to read different parts of the DNA. And the way it does that, it has to open up little bits of the chromosome. So this is a very important developmental phenomenon. However, amazingly enough, in the maize genome, only about 5% of the DNA is thought to actually encode genes. These are the 50,000 genes I was referring to before. The rest of it is often called junk DNA and is mostly composed of these transposons. So the transposons are actually part of the structure, if you like, of the way in which that book of instructions is laid out so that you can read each page one at a time. So how then do you manage to do that and then repackage the whole thing in pollen and eggs for the next generation in a way that doesn't accumulate these epigenetic mutations? Well, a very important discovery in the last 10 years has been a sort of sister of DNA called RNA. We've known about RNA for a very long time, but a particular class of RNA called small RNAs. Now, in contrast to the meter-long DNA found in a maize cell, these small RNAs are only 22 nucleotides in size. That's, actually I'm not sure what that is, it's about 200 angstroms, I think. So it's a really, really much, much smaller unit. And yet it's related, actually it is a copy of the genetic information found in the DNA. Now there are many different types of small RNAs. You don't have to remember all the details on the slide to hopefully to understand what I'm going to tell you. But some of these small RNAs operate by regulating genes. They do that actually by silencing them. They switch genes off. But others are derived, actually the vast majority of them in plants are derived from the transposons themselves. Those little pieces of DNA that can jump around. And those small RNAs are required to silence the transposons, to stop them jumping around, and to stop them making an epigenetic mess of the genome in the next generation. So these small RNAs are critical for what we call reprogramming, for taking genes, taking the genome of a plant that's already lived through, in the case of a redwood, for example, maybe a couple of thousand years, and putting that genome into the pollen so that it will go into the next generation without causing all of these epigenetic problems that we saw with clones. So it turns out that small RNA helps to reprogram the pollen in the way that I described. It's a fairly complicated procedure. We still don't understand all of it. But a couple of years ago now, we were able to show that small RNA was in fact intimately involved in this process. What I'm showing you here is a pollen grain under a microscope, fluorescent microscope. And there's a protein called Tedium-1, doesn't matter what it is, that is labeled here in yellow. And it is found in the sperm cells. Now these are the cells that are going to go on to the next generation and fertilize the egg, just like in animals. But pollen actually has another cell type. It's called a vegetated nucleus. That's shown here in red, which is actually labeling DNA. And that nucleus is not going to go into the next generation. All it's doing is protecting the sperm cells. And it's doing that by producing these small RNAs. I'm just going to summarize that. This is real data. This is now real data with a cartoon drawn on top of it. So So what we think is happening is that in the vegetative nucleus that doesn't go on, it's a little hard to see, I'll show you a better picture in a minute, you lose that DNA structure. It completely unravels. And small RNAs move from that structure into the sperm cells. Because that, of course, is the beauty of small RNAs. They're small enough to move. Imagine a meter of DNA trying to move from one cell to the next. It's never going to happen. But these small RNAs can. In fact, they can move very long distances in plants, possibly all the way up a tree. I mean, they're really quite amazing things. And so this is how we think pollen gets reprogrammed. This, of course, silences the transposons, which means that they don't cause havoc in the next generation. And that's why sexually reproducing organisms are able to reset their genomes every single generation. Well, it turns out that the same pathway works on the female side as well. I don't think it's as pretty as pollen, but on the left here is again a microscopic image of an ovule, and there's a central blob that's unstained there where the egg apparatus and the egg itself is located. On the right, more dramatically, you'll see that area highlighted in blue. The reason it's highlighted in blue is because this is now a mutant that can't make small RNAs. And if you can't make the small RNAs, the transposons come on. And that's what the blue color is. So that's showing you that transposons are silenced by small RNAs, both in the pollen and in the egg. So transposons and small RNAs are intimately related to this reprogramming business that happens each generation. Well, at the time that we were doing this research, we started a collaboration with an old friend of mine called Jean-Philippe Viel-Calzada, who's in Mexico at the National Polytechnic Institute near Puerto, and he's had a very, very long-standing interest in apomixis. That's what clonal, reproductive cloning, if you like, is called, in part because it would have a really big impact on Mexican farmers in particular, if they would be able to save their seed. And what he found was that these very same mutants actually produce clonal eggs, amazingly enough. So on the right, or on the top rather, we've got, again, a micrograph showing individual cells within the ovule, within the female part of the plant. And there's a single cell that would normally be specified to undergo meiosis, very obvious, has a huge nucleus, that will go undergo meiosis, randomly picking genes from mother and father and producing an egg. But on the left are the same thing from a mutant that can't make small RNAs, and now there are extra egg precursors now surrounding that normal meiotic cell. And these, you can show genetically, contain a complete copy of the maternal genome without any random mixing, without meiosis, any of that. These are clones. These are clonal eggs. So this is really good news. We're part of the way there. You can actually, by reprogramming plant germ cells, produce clonal, sometimes called apomictic, eggs. Unfortunately, of course, the transposons have been activated. And so these plants that are produced from this aren't that likely to survive in the long term. And so what we're trying to do now in this line of research is basically to control the transposons in some other way. And maybe if I ever come back here, I'll tell you how that's been going. So it's interesting, though, knowing what we do now, to think about why transposon silencing and what we call germ cell fate, the sex life of a plant, why they're so closely related. What I just shown you is that transposons are silenced by small RNA, but that silencing prevents clonal reproduction. And so the plant, if it wants to silence its transposons, has to do sexual reproduction instead. But why would that be? Well, it turns out that transposons actually do much better in a sexually reproducing population than they do in a clonal one. And that's because all of that random mixing of genes also randomly mixes the transposons so that they can increase in copy number. It's actually the only way they can. So one way to think about this is that transposons, sort of like the aliens in the Rocky Horror Picture Show, are forcing their hosts to have sex. And that's the link between. None of you even saw that. So that's in a sense perfectly serious. This is why transposons and small RNAs are so intimately linked with the sexual cycle of plants. All right, so enough of the hardcore science. What about the question of biofuels? I'm going to end on this fairly quickly. Okay, clones are great. Clones, naturally occurring clones, are wonderful potential hybrids that maintain the hybridity. And so, I've just explained, sugarcane is a clone, miscanthus, if it ever works out, is a clonal plant. And palm oil has a lot of uses. Maybe we can make clonal corn. All of this is good news. The trouble is that these biofuels are also food in many cases. Not miscanthus yet, but it hasn't been done. So all of these other, especially corn, and actually palm oil as well, if you start using them as biofuels, then it has a major economic impact on food prices. And we saw that actually a couple of years ago when the price of corn in Mexico went something like doubled or tripled because so much corn was being used for biofuels in the US. So we need something else. We need some other species that's not going to compete with food, which has a huge potential to make lots and lots of biomass and fix a lot of carbon. And I want to offer up to you the possibility that the smallest flowering plant in the world may actually yet save the world. It'll save itself. So duckweed, common duckweed, Lemnoideae, Lemna, it's found all over the world. It's actually the smallest flowering plants in the world. They are flowering plants. They're actually more closely related to maize, amazingly enough, than they are to Arabidopsis. You'll find them growing on fresh to brackish water in all sorts of interesting places. And their claim to fame from a biofuels point of view is that they clonally reproduce, and they do so by just budding off. And they bud off every 2 days. So they double in size. You've probably noticed this if you've seen a pond on like a golf course or something where there's a lot of fertilizer going into the water. These duckweeds can generate huge amounts of biomass in a very, very short time. In fact, here's a nice picture taken by Evan Miller from Prospect Park, giving you this idea. This is actually the peak time to see duckweed. Just go out there and you'll see mats of it floating on the surface of the water. So duckweed then has this enormous biomass potential. It produces about 20 times the amount of biomass as corn does per hectare of water, of course, in this case. But it can also produce flowers, so it has all of that sexual reproduction machinery. It has presumably it has the reprogramming machinery found in pollen. The pollen's white. It's stored in these little anthers here. It's actually really difficult to get them to flower in the lab, but Evan was able to do it. And furthermore, it has an overwintering form where instead of producing a new green frond, it produces what's called a resting frond or a turion, which accumulates 40% starch. That's like having a corn kernel on every little leaf. So it's really an amazing source of starch. And I'm not going to go into details, but we think using pathways and genes from algae that we might be able to get this starch to go into oil, to get the carbon to go into oil instead of starch. We'll see how that goes. I just want to end on a climate change note. So obviously we're hoping that biofuels are going to have some impact on carbon sequestration. But one thing we do know is that duckweeds really did do this a long time ago. So about 50 million years ago in the early Eocene, there was something like 5 or 6 times as much atmospheric carbon as there is now. So it was a really major hothouse effect. And the Arctic Ocean, because it was landlocked, had accumulated enough fresh water on the top that this started changing the climate, the ecology of the Arctic Ocean a great deal. It's thought that the surface temperature had gone up to something like 13 degrees centigrade. It's like a warm bath for some people, compared to the usual -9. But because of all this fresh water, duckweed, a form of duckweed called zova, was able to grow to huge quantities on the Arctic Ocean. And a fairly recent survey using icebreakers to go through the Arctic Ocean and drill down and get core sediments from deep enough to be able to see 50 million years ago, found that there were sediments of duckweed from 8 to 20 meters thick. So imagine how much duckweed that is. So this is, if you like, fossil fuels. And that was enough, they've done some calculations, that was enough to actually impact the amount of carbon dioxide in the atmosphere significantly and change the temperature over about the next something like 800,000 years to the next cool house that happened after that. So these plants have the capability of not only acting as a biofuel, but actually changing the climate, so long as you can grow them on the Arctic Ocean, which we probably can't, but at least we can think about that impact for the future. All right, so duckweed then maybe the answer to biofuels. I'm just going to sort of sum up what I just told you. Hybrid vigor is really important. It's probably the most important single factor in yield of any crop or any biofuel plant. Clonal hybrids can maintain that vigor because they're clones, but often they lose fertility over time, and in some cases, especially when you make them from culture and so on, they accumulate variation due to epigenetics and transposable elements, jumping genes. Clonal seeds, on the other hand, should be much more stable, and if we can make them from our favorite plants, they will be a really big help both to farmers throughout the world, but also to people seeking higher yields for things like oil palm. And clonal duckweed as an alternative to food-based biofuels, I think has a huge potential, and we're very excited about being able to just skim it off the pond in Prospect Park and throw it straight into a car. All right. And I have a lot of people to thank who we can read the names later, but thanks. I'll be happy to take some questions. Thanks.